let's go over simple diffusion and why it works. And one thing that my classes get a little bit annoyed at is that I'm always referring back to the fact that it's all about particle theory. And it's so true, because everything around us is happening because particles are moving and interacting and vibrating, and that is dependent on the state that they're in. Now, everything comes as a solid, a liquid, or a gas. We also have plasma, but we're not going to get into that in too much detail now. Solids, when you look at them, are particles that are in this sort of formation here because they have very low energy compared to their liquid and gas states. And they're vibrating, but they're not moving around because they actually can't. They haven't got enough energy to do much more than vibrate, but they still have enough energy to move in some sort of manner. Liquids are particles that have been able to break free of those cohesive forces and are slowly moving around like this and vibrating as well, and they go in all directions because there is no actual control as to what direction they move in. They just bounce around in whatever container you put them in, and as we know, liquids take the shape of the container that they're in. They will move around like this in all directions, and that's actually one of the biggest reasons why diffusion happens. Now, gas is very similar to liquid, but the particles have so much energy, they're spread right out like this. They're vibrating and moving really, really fast, again in all directions, and because of that, gases will spread quicker than liquids because they have more energy, they're moving faster, so they're going to move around quicker. Now, this little piece of knowledge can be directly applied to what's happening with particles either side of a membrane, because all of these particles are going to be in a liquid state in our cells, and of course all of them have energy and are moving. Now, here's a look at these particles up close. They are moving and vibrating in all directions, as you can see, because there is no control over their direction, and because of that, they will slowly start to change position from where they are, naturally, without any energy input required. Now, if these are small non-polar molecules like oxygen and carbon dioxide, they can literally slip through these gaps in the membranes here without having any problems. And because some of them are moving down, some of them are moving all over the place, but some of them are moving down, they're going to actually end up moving through that membrane until they're evenly dispersed. Now, because they're all on this side of the membrane to begin with and not on this side of the membrane, most of the movement across the membrane is going to be in this direction because there's nothing here to come back this direction. Once they start to move through, realize that these guys are still bouncing around, and some of them will, yes, go back up that way while some are coming through this way. But because the majority are on this side of the membrane and less are on this side, most of the movement will tend to be in this direction and less of it in this direction. In other words, they'll have an overall movement from high concentration to low concentration simply because of the number of molecules that are on either side. Call this a concentration gradient. Eventually, these particles will spread out to a point where they're even on both sides, which means, again, because they're bouncing around, some will be bouncing this way, some will be moving that way, and some will, again, be going out of the membrane as well. But because they're evenly distributed now, the amount in will be the amount out. So overall, the net movement now is going to be 0. Because there's an even number on both sides, the amount of movement in and out is going to be the same. And essentially that's how diffusion works. Because particles are naturally moving, they will end up diffusing until they're evenly spaced in an area that they are able to move around in. Now, this process works for, remember, simple and facilitated diffusion. Simple means that it's small non-polar molecules can literally just slip through the membrane like this, because they do not have any repulsion from that hydrophobic area in there. They slip between the molecules. If you're a bit bigger, or in water's case, polar, they have channels like this that literally open up small sections of the membrane for molecules to actually slip through. But in this case, you have to be of a specific shape and a specific chemical type in order to move through that. Here we have examples of channel-mediated and carrier-mediated facilitated diffusion. So here we have a channel here. Water goes through membranes like this. We have little protein channels called aquaporons, which only let water go through, and it will simply be an opening that water can fit through specifically and move pretty much unhindered backwards and forwards through the membrane. Now, not all will work like that. We also have another type of facilitated diffusion, which is called carrier-mediated. Now, carriers have a specific shape that you have to fit to, then they will change their shape to let you through. So this one here, only these little oval ones can actually fit into here. Once one slips into here, it's closed on the other side to begin with. When one's sitting in there, it will actually move, just like a revolving door, open up on the other side and let it out. But while it's doing that, the other side of it is actually shut. So these sort of proteins that actually change their shape to let things through are called carrier proteins, and it's called carrier-mediated diffusion. Now here we have active transport. This is the one that requires energy expenditure in the form of ATP, adenosine triphosphate. Now, active transport can fall into two categories. One that uses ATP directly and one that uses ATP to set up what we call a concentration gradient and facilitate a reaction later on. But we're not going to worry about that too much. When we look at this here, this is what we call a carrier protein, just like we saw before, but this time it requires an input of energy to work. Now, we often call these ion pumps. Remember, if you have a pump at all on your property or your farm where you're pumping water, it requires energy in order to move water through that pump. And this is pretty much exactly the same way. We have, this one here is called a sodium-potassium pump. It actually moves two types of ions, sodium and potassium, and it moves them in different ratios. So with this carrier-mediated protein, you actually have a little hole here that fits sodium, but it won't do anything until ATP joins up, and then it will spit sodium through from one side onto the other side. And in this case, it's made to move sodium out of the cell. You actually don't want too much sodium in the cell. Now when it does that, it opens up, and the only way it's going to go back to its original configuration is before it does that, it needs potassium to come in and sit in these two little holes here that are made to fit potassium perfectly. And once two of them sitting in there, it will split the adenosine triphosphate into ADP, adenosine diphosphate, and one lonely phosphate here. Split them using the energy, spit out potassium on the other side, and go back to its original configuration there, ready to take more sodium in. Now notice here that it spat out 3 sodium, but it only let 2 potassiums back through. This pump is actually set up so that more sodium will go out than potassium comes in. And as it quickly spits these in and out, it's setting up a concentration gradient here. More sodium will end up on the extracellular side of the cell and not so much on the inside. And on the opposite hand here, we're going to have potassium building up on the inside of the cell and not so much on the outside here. So we end up with a concentration of high sodium on the outside, low on the inside, and high potassium on the inside and low on the outside. So that's active transport with ion pumps. Remember, they're carrier-mediated proteins. The other type of active transport that requires energy is cytosis. And there's two types of cytosis, endo and exo. Essentially, endo means you take it in, exo means you spit it out. There's several types of endocytosis. Phagocytosis, which means you're eating solid particles here, taking them in. Penocytosis, which means you're eating fluid or drinking essentially. And receptor-mediated cytosis, which means something has landed onto a particular receptor to tell the cell that it's here. And as soon as it does, the membrane actually pinches off and takes that particle inside the cell. Now, cytosis is quite simple. If something is near the membrane that needs to go inside that's quite large, the membrane forms a little pocket like this to protect the cell. That large particle then gets encased in that pocket of membrane, and essentially if there's anything dangerous in here, it can't affect the rest of the cell because it's within its own membrane. That goes straight to a cell lysosome where it is joined up and it is actually eaten, essentially ripped to pieces to protect the cell against any dangerous particles coming in. Same with penocytosis. Receptor-mediated cytosis is a bit different. That's where your cell is actually waiting for a certain chemical to come into the cell, like this, and we won't go into that in detail at this point. On the off side of endo is exo. Exo means releasing things. And one of the best examples of this is our nerves. Our cells make chemicals, and a lot of those chemicals don't stay in the cells. They need to be released. And this is one case. Our nerves make little chemical signaling particles here. When one nerve needs to excite another nerve to transmit the message, these chemicals are actually taken to the end of this nerve here, released into the extracellular space. Because of particle movement, they diffuse across. And as soon as they get to the other side here, they actually hit receptors here, which induces a new message to keep on going on the new neuron. This can happen in many other cases. Say we have cells that make hormones. They're released into the bloodstream in exactly the same way. So that's exocytosis, spitting out.